Hi guys, we're going to quickly do a little overview of task 7. How does temperature and pH affect enzyme activity? So temperature and pH are two kinds of things that could make an enzyme not work anymore. Enzymes like to work in the conditions that they are made in. So if it's made in our body, it works best at the temperature of our body, the pH of our body. So we're going to see how changing that affects the enzyme's functionality. So in this case, the first part that we're going to look at is this enzyme called catalase. This is found in potatoes and as well as you. It breaks down hydrogen peroxide into water and oxygen, so into these two little gases, which we're going to see as bubbles, as foam. And it's a harmful byproduct to cellular respiration if it builds up in concentration in your cells. So what we need to do is get it out of our cells, and that's what catalase helps us do. So our question we're asking in part A is, how does changes in pH affect catalase and its ability to break down H2O? And then here's our kind of hypothesis. So if we look at these different levels of pH, pH is just how acidic or basic something is, and that's just the amount of hydrogen ions in there. And you're just going to make a prediction if you think that the foam produced is going to be greater with an acid, or is it going to be lower, or is a lower pH going to create more foam? So lower pH, more foam, or higher pH, more foam. So this is basically an overview. So you're going to have 3 test tubes. We had a little bit of potato, mashed up potato in each, hydrogen peroxide in each, and then we added acid, vinegar. We kept water as our neutral pH, and then an ammonia as the low pH one. And so basically we measured the height of each of those reactions. Remember, the more foam, the better the enzyme is working. If there is no foam, that means that the conditions were not good for this enzyme to work, and it probably got either denatured or the active site got morphed enough that it would not function in this case. So then you would go through the actual procedure, and then you're going to do your data table, graph, and identify your independent and dependent. So in this case, pH, we're talking for our independent variable would be our pH, and the dependent would be our height of the foam. Then you guys can figure out the other 2 parts. And we'll do the same thing, but this time we're talking about temperature. So your hypothesis is exactly the same, except where you're trading out pH for temperature. Same thing for your hypothesis. Again, you're just looking at, at that enzyme function. And so here's kind of the overview for this one. Again, potato and hydrogen peroxide. That's all we put in these tubes. All we have now is then we place the whole test tube in hot water, a hot water bath. This one we just left in the test tube rack, the room temperature one. And then the cold water, we stuck it in a beaker of cold water to see how much foam was produced. Again, more foam means the enzyme is working, and we're seeing it being functional. Here is some example data for you. So you can use this in your lab to complete it. And if you have any questions, please let me know. Then you just kind of finish out the lab and make sure that you turn it in to me. Okay, if you have any questions, give me an email.